Okay, connect the computer audio. The audio, it's the speaker, yeah. Okay.
You're already in.
Yep, in the PowerPoint.
And so if I now make you I'm going to make you host. You are now host, Didac,
Okay.
of the Zoom. The things you will need to know.
Okay. Just put it in this, or just put it.
Nobody saw this.
On the top. Okay. And pointer. Okay, it's a point. No, I mean the point is, yeah, it's there. Just if you move it, yeah. Okay, but I'll always do. Okay, it will be like a game. Like here it is. Okay, so what else?
Hello.
Hi there.
How are you?
I'm here preparing.
Organizing things. Yeah. That's why I come earlier in case it's help.
Yeah, because you've heard about this. Soutenance is going to be a bit strange because it's all, and none of the committee can be here, so it's all virtual.
Yeah.
So I cannot be here. I have to now go and do it by Visio from INRA.
Ah, okay.
So there is going to be no one here. Sandrine Petit will do some help, but it's going to be a bit strange for a little while.
There is something else we need to no, just wait for the jury to connect.
Yes. So you might need to let them in.
Okay.
You're the host. There are two people on here at the moment. There's yours.
You and me.
Yeah.
Okay.
Yeah.
And
so your sound is going on at the moment. You can cut that for a minute.
this camera work like this. If not, I can use.
It's the problem with this camera. Is the camera on the actual Macintosh one? Because there's a camera here.
Okay. Normally no, but I can use this camera and just let it.
The problem with it is it's all one of these 180, so it can do all the way around, and it jumps around based on the movement.
Well, it can be funny because I will be moving. So
Okay, I'm going to go
okay.
because I'm starting about 10 minutes. Good luck. I'll be seeing you in about 2.5 hours, 3 hours.
Hopefully.
Not hopefully, yes.
Yeah, good or no. I'll go to the telephone second. Okay.
Coucou, entre, vas-y. Il n'y a personne. Ça va, ta soutenance s'est bien passée?
Oui,
Cool,
ouais.
c'est bien. T'es débarrassé.
Je
C'est ce que tu vas faire là?
vais faire un projet avec un chercheur. On a défini les objectifs, donc
Bon courage.
Et toi, comment ça se passe?
Moi, je me suis réinscrit pour
D'accord.
l'année prochaine. Du coup, je vais soumettre mon manuscrit en janvier, début février. Et ensuite, j'aurai une soutenance soit fin mars, soit début avril. Voilà. Parce que j'étais en arrêt vendredi.
Ah oui, quand même.
Ouais.
Hi, you're a doctor?
Yeah.
Congratulations.
I'm sorry I didn't come to the test because I had to do a repetition of this and I didn't pass and I wasn't able to come.
No problem, I understand.
How was it? The questioning and this was okay?
At the beginning, the first one, I was really, destabilized.
Yeah.
So I was like, he asked the same question, but rephrased it every time. So I was like, so I didn't answer well because he asked the same question. So I was like, I don't know what you want, what do you want? But at the end, it went well. It was better after the first one, where I was destabilized, it was better after. And they congratulate me, so it means that I answer at least.
How much time it was?
Not that long, I think it was 1 hour question. Not that long. I know. Especially I didn't get a lot like.
Yeah, I
I
understand.
will turn off the camera because people is seeing us.
Okay.
I can you see me? Because
Yes.
okay,
Can you hear me?
yeah. 1 second, the volume. Okay, now better, I think. This guy with the microphone and the camera never appears, but
Okay.
we have these ones, so I think it would be enough.
Everyone in the audience looks a little bit like they're sort of drifting off the page, but
Yeah, I can try. Maybe do it need something like be better?
cool. Right.
Yeah,
Hello, Didac.
hello. I'll go to close the door 1 second. Okay.
Okay, is everything okay at your end then, Didac?
Yeah, I'm here. I don't know if you see me because I don't see my camera. I don't
Yeah,
know.
no, we can see you. And as you move around, I can still hear you, which is the important thing.
Okay. So we can start when you want.
Okay. All right then. Good afternoon, everybody. I would like to officially call this Viva to order. So please welcome all to the PhD defense of Didac Barroso-Bergada. I hope I pronounced your name reasonably correctly there, Didac.
Perfect.
Joining us today for this defense, the committee will comprise 2 rapporteurs, myself and Christophe Mugel, 2 examiners, Lucie Zigna and Tristan Cordier, and also present is Didac's PhD advisor, David Bowen. My role in this is to act as the president and make sure that the Viva runs to order. And shortly I will ask each of the panel members present to introduce themselves so that you're aware of who is who and who will be speaking to you today. And then after that, we will invite you, Didac, to present to us your thoughts on your thesis. You've got about 40 to 45 minutes to do that. And then we will ask you a series of questions, starting with your examiners first, followed by questions from your rapporteurs. Is that all reasonably clear? And importantly, have you heard everything I've said?
Yeah, perfect.
Great, that's wonderful. Okay, so I'd like everybody to introduce themselves now. I'll start off. My name is Alex Dumbrell. I'm a molecular ecologist from the University of Essex in the UK, and I'll be acting today as the chair of this Viva. I would then like to ask Christophe if you could introduce yourself.
Dijon?
Oh, I can hear Christophe.
Try again something.
We have some audio problems, I think, from here at least.
It's just dropped out again, Christophe.
And now it's working out?
Yep.
Yes, do you hear me? Sorry. To move from different platform, it's each time a big problem for me. So hello Didac, hello everyone in Dijon. So sorry to not be present in Dijon for your defense. So it's a pleasure to be the reporter of your PhD. And so I working most on microbial ecology and plant microbiota interaction in the rhizosphere, and try to understand this interaction and the role in plant defense or plant adaptation to different environmental stresses. That's it for me.
Brilliant. Thank you, Christophe. Lucie?
So thank you. Hi, Didac, and thanks for the invitation. So my name is Lucie Zinger. I'm an assistant professor at ENS in Paris, and I'm specialized on environmental DNA techniques and macroecology of microbes in the ocean or in the soils, and I also work now on invertebrates.
Thank you. Tristan?
Hi, Didac. Thanks for the invitation too. So I'm Tristan Cordier. I'm a senior researcher at NORSE. It's in Bergen, Norway. And I'm also a molecular ecologist working with environmental DNA, mostly with marine sediments. and I work also on developing DNA for biomonitoring application.
That's great. Thank you, Tristan. Okay, so Didac, in a second, I'll ask you to start by presenting some of your thesis thoughts to us. I will say to everybody that it's perfectly okay for questions and or answers to be provided in both English and French, and the choices of the asker of the questions and the answer of the questions to choose which language they use. That's entirely acceptable. So over to you, Didac, please begin.
I will share my screen. Okay, and I will, 1 second, try to hide the here. Okay, here. Sorry, I'm preparing everything. Okay, do you see the slides presentation there?
Perfect.
Okay, so thank you, Alex. And yeah, I will start the presentation. I will present the work I did during my thesis in automatic learning of interaction networks from next generation sequence data. I'll take the pointer. So why interaction networks? Well, I did my thesis on the frame of the next generation biomonitoring project. And the aim of this project was biomonitoring project was monitoring using interaction networks. We decided to use interaction networks because when you do biomonitoring, you can start monitoring the different factors like temperature, precipitation, etc. that can affect the ecosystems. You can also start monitoring the different species that you have, so you can see if they are present, absent, how abundant they are. But if you want to be able to be sure to know how the ecosystems behave, what you need to know also is how the different organisms interact between them. And for that, we use interaction networks. So an example on how to use an interaction network to biomonitor, it's here, the one we can see here. We have a context of climate change where the sea ice is retreating, so the water is getting warmer. And we have an Arctic food web of fish, in this case, that it changes because a new predator enters. So if we're able to understand how the interaction network between the different fishes changes, we will be able to see how the ecosystem change, but also to predict how it will change in the future and take measures to avoid the change or to at least limit its effects. So during this presentation, I will talk a lot about interaction networks. What is an interaction network? Well, interaction network is a graphical object that can be quite simple or be really complex, but it has two main things. It has nodes, where in the case of ecology, we have the species, and it has edges that symbolize interaction. Then it can get much more complex. We can have different sizes of color of the nodes depending on phylogeny or abundance. We can have different colors or size of edges depending on the strength of interaction or on the type of interaction. To infer these interaction networks during this presentation, I will talk about two main strategies. And it would be good to have it clear the difference between them. They are correlation and logic-based network inference. For pointing the differences between correlation and logic, I will put I'd like to use this example. Here we have the Rosetta Stone. I think most of you know. The Rosetta Stone was used to translate from ancient Egypt, which was an unknown language, to Greek. So, and then from Greek to any other languages. And this was done by a French guy called Champollion. To do this translation, nowadays, what we would do is a correlation-based strategy with a translator available like DeepL or Google Translate. With these tools, what we will use, it was just correlate the position of the words in Greek and the words in ancient Egypt. So we assess how normally which word are correlated with which and how significant it is. So we will assign a translation between Greek, direct translation between Greek and Egyptian. This now is quite useful, and we use it a lot in our daily life. Me here living in French, I do it a lot. But this is the mechanism that does not offer information to us on how it's done. So the machine does something, and it gives us a really interesting result, but it's a black box what's happening. It establishes correlations that we don't really understand between words and offers us a translation. This would be the correlation strategy. If we think on the logic strategy, it gets closer to what Champollion did. Champollion knew grammar and knew things about ancient Egypt. So he used his knowledge on grammar and ancient Egypt to translate. So he started studying putting first some grammar rules, try to see how the different languages were following these grammar rules, and then using it to translate. For example, he knew that there are phrases in a language, that in each sentence in a language, we have a verb, we have a noun, and we have complements. And they are structured following an order in a language. And then from there, you can compartmentalize the different sets of the text. And from there, you can start translating, figure out, okay, here I have a sentence, here I have sentences, this is the verb of the sentence, this is the verb, this translate like that. This process is also, you can be used for translation, and it's understandable. So it follows a logic process that uses rules to translate in what we call explainable machine learning process, if we use machine to do that instead of our brain. How this translate to ecology? Well, the grammar rules are not useful for ecology, but we can have rules in ecology. For example, there are really basic rules in ecology. To have an interaction between any two species, we need them to be together. Or in the case of arthropods, for example, normally the big guy eats the small guy. Or to eat different kind of animals, you need to have a mouth adapted to eat them. So we can infer networks using logic, just relating these ideas and checking how this affects the different members of the community, and we can construct a food web, where we can also use, for example, the abundant information that we can obtain from the different members of the community, correlate them, assess which correlations are positive or negative, and have an interaction. So after explaining a bit this difference between logic and correlation, because it can get a bit complicated later, I will do this order of presentation. First, I will tell you how we obtain the ecological data to infer networks. So how we obtain the information we need to assess how the different species interact. Then we will test some available correlation-based tools to infer networks for monitoring. And then I will describe the process I followed to develop a logic-based application to infer interactions. It would be in 2 parts because the process was long to develop this, and we did some first tries and we tested them, and then we improved our system to end in a tool that we call Infinite, that it's called runner package. So first, how we obtain the data. During this thesis, I focused on microbial interactions. Microbial interactions are interesting because the microbes are everywhere. In each ecosystem, we have microbes, and they do really important stuff. Depending on the ecosystem, they can provide nitrogen, they can provide support to the development of plants, for example. But for example, they also can be pathogens. So they are involved in many ecological processes interesting. But the problem with the microorganisms is that, as you can see in these images, they are really difficult to observe. It's really difficult to study how they interact by observation or by other techniques. But nowadays, we have the DNA that allow us to obtain information about these microbes, and we can start inferring the interactions that we have between them. How we obtain this information? Well, some of you talked about metabarcoding when you were presenting yourself. Metabarcoding is a process that allows us to use eDNA, ecological DNA, so eDNA that we sample from an ecological context. In this case, it's a leaf. On the surface of a leaf, there are many microorganisms living. So we can sample the DNA on this leaf and sequence the DNA present there. Each species has different DNA, and some regions of the DNA are really useful to differentiate the different species present in our sample. So if we sequence explicit regions of the DNA that we call that we use them as a barcode, we can identify different species that we have on our leaf, for example. But we can go further. When we sequence, we obtain a list of sequence. So what we can do with this list of sequences is try to join them in what we call operational taxonomic units, join them together by their similarity, and all the sequence, the count of the sequence joined together would be a measure of abundance of the different species that we have present. During this presentation, normally what we have information about is all to use, but I will use the word species to be more clear. There is another way of doing it, of producing operational taxonomic units, that it's inferring ampli-consequence variants. The difference between O2Us and ASVs is that to produce ASVs, instead of just cluster the sequence by similarity, we study which sequences could be the same if we take into account the errors that can exist during sequencing. But the idea is the same, is having a group of sequences, so a measure of the abundance of the different species present, that give us an idea on how abundant they are in the system. And when we have enough samples and we know the abundance of different species along the different samples, we can start inferring interaction networks. How do we infer these interaction networks? Well, we will follow this process. First, we sample the DNA from our leaf. We amplify the DNA for our barcoding region. We sequence. We then cluster this sequence, producing these O2Us or ASVs. So we have the measure of the abundance, and we then reconstruct the network. To reconstruct the network, we can use correlation tools, as I say. How the correlation tools work to reconstruct the network? There are many correlation tools available now, if you're interested. But the general idea of it is, here we have an image of the network with just 4 species, and we know the abundance of all the species along the different samples. What these tools do is just study the correlation that exists between the abundance of the different species along the different samples, and then assess if this correlation is significative. And the correlation that we find significative is the one that we make an association between the 2 species involved. This can get really complicated, and there are many methods to do this. So there is pure correlation. We can use other measures of like inverse covariance that has into account also the abiotic effects. So this can get really complicated. But the basic idea is this: is establish a correlation or a partial correlation between the different abundance of the species, and then associate them. So having these tools available, what I did during my thesis was extend the work that another PhD student did, Charlie Covert, to test 2 of the most used tools in the literature, SparseC and SpeakEasy. I will be referring to both during the presentation. They both are correlation tools for doing correlation networks. So I tested these 2 correlation tools to produce networks. And with these correlation tools, what we can produce are association networks of positive and negative significant correlation. So to do the testing, what I did was saying, okay, we need to use these tools for biomonitoring ecosystems. For biomonitoring ecosystems, we're interested in detecting ecosystem change. So we took an ecosystem that changes. It was the change between using conventional and organic cropping system. So our conventional organic cropping system uses different pesticides, uses different methodologies to expect that the interaction networks between the 2 cropping systems would be different. So we expect that our tools, or correlation tools, SparseC and SpeakEasy, would be able to detect the differences between these 2 cropping system networks. But also we expect that if we use the same cropping system, the networks that we produce from the same cropping system would be similar. So we did this test. The experimental design was this one. We got 3 plots of grapevines, organic, and 3 conventional grapevine plots. We sampled the leaf of 20 vines in each plot, And we did all this metabar coding process, so sampling, sequencing, amplifying, and clustering using a really broadly used pipeline for ISV clustering called Didac. Then, to be able to see if the network produced were different or were the same, we needed to have a measure on how similar or different are the networks. So we take a look at the available measures for networks. So we have network alpha properties, which are the characteristic inherent to the network, like the number of links, the diameter of the network, the connectance of the different nodes of the network. And we have the network beta properties that are the ones that we obtain when we compare two networks. And what we saw with all these measures, it was that only two of these measures, the overall dissimilarity of associations and the dissimilarity of associations between shared ISVs, were able to capture a difference between the networks from organic crops and the network from conventional crops. So here we have the results. This is a bit complex, but we have a panel for sparsity and a panel for speakeasy. What we can see on the top left would be the interaction networks that we get, so 6 interaction networks. And then here it's plotting, decreasing the number of ISVs used for plotting the network. Then on the VE panel on the right side, we can see the different alpha diversity measures. As you can see there is no difference between conventional and organic crop. But if you use the beta diversity measures I mentioned before, we can see that we can cluster the different networks that we obtain by being either organic or conventional. Then we check if effectively the networks that we were producing using the same cropping system were similar. And what we found is that even if the networks had several hundred of species and several hundred of interactions or associations between its members, we only had 5 common associations between the 3 replicate networks. So what we saw that is even if we are able to capture the ecosystem change, it's really difficult to produce similar networks from the same conditions. This has many reasons, can be caused by the stochasticity of the system, by the different biases introduced during sampling, etc. So as a conclusion of this first study of correlation measures to be used in biomonitoring and correlation tools, we saw that alpha properties do not differentiate between cropping systems, but some beta properties do. We saw that networks inferred from replicate cropping systems were not really similar and had few concentric associations. And when we started to look at the networks and try to figure out what was the ecology behind, we also saw that we were obtaining networks with this positive and negative correlation, positive and negative association. But if we try to understand the ecology between these interactions, it gets a bit complicated because a positive or negative correlation can mean many things. For example, if we have a positive association between 2 species, it can mean they are in a mutualist interaction. But it can be also a commensalist interaction or even that they have the same abiotic preferences. While we have a negative association, it can be that we have a predation interaction, but also a competition interaction or different requirements for abiotic factors. So if we think in the paradigm that we are using when we use correlation tools to produce networks, we have this. We have our abundance information that we obtain from correlation tools. We then use these tools, sparsity and speakeasy in our case, to produce the networks. Then we obtain a list of networks with associations between the different species. Then we need a microbiologist or ecologist that comes here, looks at the network, and it starts figuring out, okay, this network is really complex, but this link could be competition because I know that these 2 species need the same organic compound to develop, for example, but needs an interpretation, and this interpretation can be subjective because an ecologist or a microbiologist does not know everything, and he has his own interest, etc. So what we try to do is move to a paradigm like this, where we have the abundance information, and if you remember with the Champollion or example of the arthropods, we put some rules for interactions before the inference. And we can also introduce other knowledge from databases like functional information from the species. And we use these rules of the abundance information to infer directly the interactions so that we avoid the interpretation stage. To do this, first we need to figure out a rule for what a microbial interaction is. So if we imagine two different microbial species, we can see that a potential interaction between them is something that causes an effect on the other one. But since the information that we have about this microorganism is abundance, the rule would be that an interaction for us is any effect on one species on another that causes a change on the abundance. But this change on the abundance can be positive and negative, and we can have many combinations between both of them. So at the end, we have a general rule for interaction. An interaction is something that causes a change on the abundance. And then we can have a list of interactions. So we can have mutualism interaction. That means that both species are in an interaction that increases their abundance. But we can be in a competition interaction, but it's the opposite as mutualism. We can have predation or parasitism, where one increases, the other decreases. Commensalism, when one is favored, so the abundance of one increases while the other one is not affected. And the opposite to commensalism, that would be amensalism, that one species is not affected and the other has its abundance decreases. So now we have a rule of interaction and an idea on how then to classify interactions. That we need to decide what's the logic process that we will follow for inferring interactions. The most famous logic process and we are more familiar with is deduction. Deduction is a logic process when we know a rule. In our case, that an interaction produces a change in the abundance, and we see something happening to taxa interacting. So this is the information we have. And following this logic process, we can infer that the abundance of at least one of the taxa will change. But if we see our case, we are not in this paradigm. What we have is the rules of interaction and the result of this interaction, but we don't know which interaction is happening. To this, we have to select another logical process. We have other logical process. We have induction, which is the case where we know what's happening and we see the result on what's happening, and we can start establishing rules for what's happening. Or we have the process that it will be the one we follow, which is abduction. It's a process where we know the result, the change in the abundance. We can get this information from our meta-barcoding data, and we have our rules of interactions. And what we will abuse is what's happened to produce these changes in abundance, so the interactions. How we follow this abduction process? First, we have the information, the abundance information from meta-barcoding. So here on this table, we have different species and the abundance of the different species in the samples. And we need to express this information in a logic clause. This logic clause has this format. We have the abundance of species 2 between sample X and sample Y that goes down, as you can see here, between here and here. And for example, we have the presence of a species 1 in sample X that it's no, that means that it's not present because its abundance is 0. And if it's present, because it's more than 0, will be a yes. Then we need to express all rules relating this information that we have. So we have a lot of observations of abundance change, and we need to relate them using logic rules. As we said, the interactions are based on the effects on the abundance, and we can have effects increasing the abundance and effects decreasing the abundance. So our relation between these concepts would be that there is an effect up on species 1 on species 2 if the abundance of species 2 increases when species 1 is present in the first sample and not present in the second sample where the increase or decrease happening. And the same thing can happen as opposite for an effect down. So there will be an effect down if a species 1 is present in sample X and a species 1 is not present in sample Y. Then we do the abduction. What abduction does is use all the observations that we transform in logic clauses to look at them and figure out how many of these observations support each possible effect on all these different species. And we get a measure of this information that we call compression. So compression, we have to understand them as a measure of the amount of information that we have on our metabolic coding data supporting if of each rule. And then we will have a compression measure for the effect up on a species 1 and on a species 2, and for effect down of a species 1 on a species 2, and any other combination of species. But if we think about it, for an interaction to happen, not only has to be an effect on the abundance. This effect on the abundance has to be constant. So in our description of the interaction, it is not possible that sometimes an interaction causes an increase on the abundance and sometimes causes a decrease. No, the direction of this change on the abundance has to be constant. So what we do is we draw the effect up and the effect down that we obtain for each combination of species. And at the end, we have a measure of how likely an interaction is, a numeric measure of the abundance that we call I statistic. So once we have this framework, we started developing and testing this logic framework for inferring interactions. For testing, we used computer-generated datasets. Why you use computer? These datasets were published before by Vegetal. Why you use these computer-generated datasets? Because there is no microbial dataset that we know enough interactions happening in them and we can use for benchmarking. So all the microorganism datasets that we have available have been obtained in multiple ecological contexts, but we don't really know what are the interactions happening in these data. This simulation that we decided to use from Bayes et al. Had the good point that it was first simulating the abundance of the different species, how to use, and then introducing interactions by changing the abundance following the mechanism of different types of interactions, like commensalism, competition, or amends. This simulation of the interaction could be modulated by a strength of interaction. So we could have a weak interaction with a 2 and a stronger interaction with a 5. And we also could decide the number of samples that we use for the testing. In this case, we selected 50 samples. Then with this dataset, we could perform the adduction. For the adduction, we used a previously designed program called Progol. It had some years, but it was able to use all these logical statements or rules of interaction to give us the compression measure. And then with this compression measure, we could obtain the I statistic. And then we needed to select which interactions were significant. So to do that, we did a bootstrapping procedure, resampling all the abundance clauses that we had to try to see if the interactions that we were getting were significant or no. And we tested, and as you can see on the tables, in the column of logic, we can see that our methodology, bootstrapping the I statistic, had a good accuracy for detecting interactions, some 97% for the weaker strength of interaction and a bit less for the strongest kind of interaction. And this accuracy was comparable to SparseC, that if you remember, is one of the tools that I used before on the previous experiment. So we saw that our methodology were able to have an accuracy comparable to to a correlation-based tool. And interestingly, we also saw that the accuracy detecting interactions decreased when the interaction was stronger. This is a bit anti-intuitive because you should expect that as interaction gets stronger, you will see it easily. But the problem here is that we saw that we were not modeling a case of exclusion. So when the interaction were strong enough, we were decreasing the abundance of the different species to 0, meaning that they were no longer present in the system. And our rules of interaction were not having into account this ecological procedure. So when the interactions were producing exclusion, we were having less accuracy. So the first conclusion we saw testing this first development of the logic-based network inference, it was that bootstrapping the I statistic, we could have this accuracy comparable to SparseC, and we saw that the strength of interaction had an effect because of exclusion. And we also see in the more mechanistic part, it was that the abduction process was kind of slow. So it took days to be able to abuse a not really big abundance matrix. So we tried to improve. The first step for improving was trying to move from this program PROGOL to another program that allows us to be faster. And after discussing with our colleagues in Surrey University, which are specialized in logic machine learning, they developed a new program called PyGol, which is able to do the same thing that PROGOL was doing, but much faster. So this program had a development process, but after it was possible to perform the abuse, the same abuse that abduction that we were doing with PROGOL, with this PyGol program, much more fast, having similar accuracy. And we also decided to move from bootstrapping the interactions to another selection process called STARS, which is based on the stability of the links that you have on the network based on how you change the ISTAC threshold around different subsets of the data. And this methodology is focused on networks, so it can give you more accuracy sometimes, but also needs less resampling, so you can even go even faster. So with these changes, we developed our tool that we call Infinite, Inference of Interaction using explainable machine learning. This tool is able to directly from the abundance data that we have from metabarcoding, transform it to logical clauses that are examples of the changes of our abundance matrix, use our hypothesis of interaction to do the abduction and obtain the effects, then use stars to select the effects that were significant, and then, since we know the direction of the effects, directly classify the interaction by the types I described before. So we tested this tool. To test this tool, first we used the same datasets we were using before, the ones from Weiss et al. That had different strengths of interaction and were simulating for different kinds of interaction: commensalism, competition, mutualism, or isomensalism. But the aim of this tool is doing it by monitoring, so we need to test it with real data. So we also test its performance using a metabarcoding fungi dataset from grapevine crops. In this case, there was no organic or conventional cropping. In this case, there were 9 vineyards from around France suffering from a pandemic of downy mildew caused by Plasmopara viticola. And we were really interested and see how our tool was performing, but also see how our tool was performing at different sizes of information. So depending on the number of samples. So what we did was produce tables following the methodology of Weiss et al. That had 20 samples, 30 samples, 40 samples, etc., until 90 samples, to see which was the number of samples that we needed to have for our tool to perform well. And we compared the performance of our tool, the accuracy of detection, with the other tools that we tested before, SparseC and Speakeasy, that were, as you remember, the correlation-based tools. And we can see that when we have few samples, like 20, our tool had less accuracy, but when the number of samples gets to between 40, 50, the accuracy is comparable to the other methodologies. And as we increase the number of samples from 60, there is a plateau that shows that no better accuracy is get from these tools. Then we were also really interested to see on how the different interaction types affected our accuracy. For example, here we can see how many of the mutualist interactions or simulated interactions were founding. So imagine that this dot line is the 100% of mutualist interactions detected. Here we can see that with infinity modeling exclusion, we were able to detect around 80% of the mutualist interaction. And we saw that depending on the interaction type, we have different accuracy. We weren't able to detect any commensalist links. We were performing good with commensalism, and we were not so good detecting competition. If we compare these results with the ones of the Speakeasy, which is the correlation-based tool, we see that they are similar. Similar in the way that for these mutualist and commensalist interactions, we are detecting similar amounts. We are all not detecting commensalism, and we have big problems detecting competition. Even if here Speakeasy seems to perform a bit better, mostly with competition. But what with these correlation tools we cannot do, and Infinity allows us, is then try to these links that they detect as positive or negative correlation, directly classify them for interaction. And we can see here, following the coloring, that the mutualist interactions that we detected, most of them we classified them as mutualism, while only a small percentage were classified as commensalism, and similarly for the other interactions. This shows that even if we can have problems detecting interactions, so depending on interaction type can be a bit complicated, the interactions that we detect, we classify them correctly. So when we detect a competition or a commensalism interaction, we can classify them as competition or commensalism, we can be quite sure that is correctly classified. And with this information, we move to the real data. As I said, this real data was obtained from 9 different vineyards around France and was suffering for a pathogen, which is Plasmopara viticola. Here we can see the measured concentration of the pathogen on the asymptomatic samples and the symptomatic samples. We collected both to generate a dataset. And we saw that obviously when there was a symptomatic sample, we had more pathogen. And then we also saw that an important factor structuring the data, if we do an ordination of the different samples, of the communities of the different samples, is that if you see here the colors on the map, blue, purple, and yellow, so which are the 3 different regions where we had the vineyards, we can see that it is the most structuring factor. So we constructed the network from these real datasets. Here there are, for example, 3 networks from 3 different vineyards we call ABC. But and we see that the amount of interactions of each type that we detected is similar. It can be similar between vineyards, but it's not the same for all the interaction types. For example, here we can see that the most important interactions by amount were commensalism and competition mostly, while predation or parasitism was almost not detected. But this does not confirm us that our methodology works with real data. What we did to confirm that we were doing good with these real networks was focused in a small part of this network, which is, as I said, we have a pathogen called Plasmopara viticola. It has a great economic importance because it causes a lot of economic loss, so it has been really studied. So we don't have a lot of information about all the interactions happening on the network, but if we focus on the interaction that Plasmopara viticola can have to other microorganisms to control its growth, so this kind of competition or commensalism interaction, we can have a lot of information on the bibliography. So we focus in this subnetwork that we can see here on the table. Here we have the fungi species, the list of the fungi species having a negative interaction with Plasmopara, either competition or commensalism. And we listed these species and searched on the bibliography if they were described previously as Plasmopara viticola antagonists. What we can see here is that 6 of them were already described as antagonists of Plasmopara. And if you check on the bibliography, these are mostly all the antagonists found. So all the antagonists that were present in our dataset and on the bibliography, we found them. So it's good, we found it what we were expecting. And for most of the other ones that were not antagonists of Plasmopara, we found that they were antagonists of other pathogens, mostly in grapevine also. So it means that, okay, they have never been described as Plasmopara antagonists, but they have the potential to have an antagonist effect on the pathogen. And we also had 3 species that we found to be antagonists, but we never find anything in the bibliography. So this can mean that they can be candidates to be antagonists of Plasmopara viticola. So we were fairly happy with our Infinity tool because it was able to perform as good as the correlation-based tools. It was able to classify interactions and was performing good with the real data. So we called it an R package, which is easy to install, which is an GitHub repository, and our aim is it can be used by ecologists. So we also wrote a tutorial vignette to teach how to use it, which is kind of easy because with a function you can go from the abundance matrix to obtaining your network. And we complemented this tool with an interactive application to visualize networks. Because networks are really complex, as you've seen before. They can have many nodes, many edges, and you need something interactive that helps you to visualize. We checked on what was available on the internet, and we were not happy because it was not responding all our needs. So we developed this tool that allows you to plot an interaction network, select subnetworks, move the nodes around, and put different thresholds on the network, and color by interaction types. So, okay, during this talk, I've been talking about correlation-based tools and logic-based tools. So, if somebody wants to do biomonitoring, what we should choose? Well, correlation-based tools has the pros we've seen. They are really robust mathematically and can take into account composition or abiotic effects, require smaller sample size than infinity, and they are fast. And also the other good point is there are many papers about them, so we have references on the bibliography about results of these tools. The cons is that the networks that we obtain, the association the networks that we obtain using them require interpretation. And they don't have the flexibility to introduce these interaction rules to the inference or other ways of background knowledge, such as the one that we can obtain from databases. While our tool can detect and classify interaction types with an accuracy comparable to correlation methods, we can reconstruct interaction networks from either synthetic or real data and different hypotheses of interaction. We can use knowledge that we obtain from databases, and everything. We can use everything that we can explain as logic clauses and relate it logically with the other information that we have. The problems that Infinite has is that it needs a larger sampling size, and even if we improved a lot the execution time, it still takes more time than correlation-based tool, but we are working on it. So if we want to do biomonitoring of an ecosystem, here we have an ecosystem. This is a river, and we have 3 different parts of the river, and we want to do biomonitoring of this river. We can do many things. We can measure the temperature of the water of the river. It's a way of monitoring it. We can do metabarcoding on the samples from the water of the river, so we will know which species are in our system. This is also a monitoring. We can use this information abundant information that we obtain from metabarcoding to use correlation tools to produce some networks. And we will have these association networks at the different parts of the river. So we can start to study how the networks of the river change along the different parts of the river, or if from one season to the other, there are changes on the interaction network. Or if we're interested on what are the interactions happening in our system and how they can affect different ecological process, for example, oxygenation of the water or some kind of sediment decomposition, then we can use the logic-based approach to start construction networks where we see which interactions is which and start understanding the ecology behind our networks. So there is no better tool, a tool better than other, or a method better than other, but all of them are useful depending on the kind of biomonitoring we want to do. So to end, as a conclusion of this PhD, we got Infinite. This is our tool, interaction inference of interactions using explainable machine learning. And we also tested the correlation-based tools, and we identified some network measures that are useful for biomonitoring. So we have created a framework that we think that it's really useful to start doing biomonitoring. So we have the tools and we have the measures to start studying the ecosystems. And in the future, I would like to keep working on this Infinite tool, introducing background knowledge from databases, our new interaction hypothesis. I would like it to start being used by ecologists so I can do testing, so improve the tool. And at the end, the final aim is to be able to automatically biomonitor ecosystems. So directly we can sample the DNA on an ecosystem, do all the process until we get the network, start studying the network, seeing what's changing, and taking managerial decisions to avoid ecosystem loss. And that's all. Thank you for your attention. I would especially I would like to especially thank my supervisor, Dave, and also the people who I work with during this thesis, Ali Reza, Corinne, Nika, and Danny, and the members of the jury, and everybody from the INRA here in Dijon that made me feel good living in France. Thank you.
Thank you, Didac. That was a lovely talk, very interesting to hear. So shortly, I'll ask the examiners to start their round of questioning. They'll speak each for about 15 to 20 minutes and then move on to the next one. Yeah, you got a drink of water, you're ready to go? Yeah, brilliant. Okay, so on my screen, first up is Lucy. So please, Lucy, begin your questions.
Okay, so first of all, thanks a lot, Didac, for your manuscript and for your talk, which was really interesting to read and to hear. And I really enjoyed, reading your manuscript because it discussed about a new method that, in my sense, is much more conceptually more convincing, in terms of how you could infer ecological networks from any kind of ecological data. So for this, it was really nice to see. I found that the work was really serious. I mean, it was done, really in a straightforward and robust way. And yeah, so thanks again for that, and congratulations also for all the work you've been doing so far. So I'm not here only to congratulate you. I have also a couple of questions and remarks about your work. So just in terms of remarks, first regarding your manuscript, I found sometimes that your state of the art was a bit light in the sense that sometimes I wanted to see more about what we know really in terms of microbial interaction, if you can give some examples and these kind of things, because I thought that it was missing somehow in the text. And also regarding environmental DNA, I found sometimes that the definition were a bit missing or not always super accurate. For example, I did not find any clear definition of what is a barcode. So it's just details, but I think it's something that you might improve in the future in your different works. Now, so but again, it's more cosmetic. Huh? So now back to the whole work. I was wondering, so conceptually, you were somehow very ambitious. Huh? You have three main pieces of issues you want to tackle. So the first one is inference of interaction networks from complex systems, basically. The first one, the second one, sorry, is the fact that you are looking at microbial communities for which we have very poor knowledge in terms of interactions. And on the top of that, you add the environmental DNA and metabarcoding world, which has several uncertainties. And so, yeah, I was wondering, to what extent how you could simplify a bit the problematic. So how whether you consider, for example, to not look at microbial systems but other systems, to try to validate your tool, your inference tool, for example.
Actually, yeah, one of the things we want to do is Dave, my supervisor, has some nice data sets of arthropods that we can potentially use for this validation. But I focus in microbial data because I think it was interesting, particularly because of that, because this environmental DNA is the only way we have to obtain information from there. So developing this DNA methodology was the only way to study microbial interactions. And for testing, yeah, we have this problem, but one of the solutions is the when you say we have some data sets of carabids, well, Dave has some data sets of carabids, and you have the abundance information of the carabids, but you also know the sequence what it's on their belly, so you know what they are eating. So you can have some kind of validation here. But yeah, in general, that's the main problem working with the microbial world is validation in all cases using eDNA. So maybe there is people who work in more mock data sets that are grown in culture. Maybe it could be another option. Try to simplify things, start working with small mock communities, try to see if we are able to detect the interactions that we expect from there and go bigger. But again, you have the problem that these mock communities does not represent an ecological reality necessarily. So you can also be cheating with it. It's really complicated for me. It was at the beginning really complicated to find a basis where you can have something that you can be sure of. We use these computer-generated datasets, for example, but yeah, validation is the main problem here.
And so, but I was wondering whether, you know, because to validate your tool, you use like data from fungal communities.
Yeah.
Which is, I mean, we don't know really, you know, a lot about the type of interactions. And I was wondering whether if you would have, you know, use a data set that encompass a bit more trophic levels, for example, and it can be done with, for example, plankton communities. So in interocean data, for example, you have several trophic levels. Maybe you might have more expectations, you know, on the type of interaction that could occur in your system, and hence, you know, you might, you know, be a little bit less in the like the uncertainty of what you should expect.
It's a really good suggestion. It would be really interesting, and also I think that it's really interesting, and maybe a paper just on that, on providing some good testing datasets. I think if a paper providing a good dataset for testing other tools or doing benchmarking can be really interesting. Not having it talk on my tool, but in general, to just have this, which is this plankton that generate this plankton dataset where you really are describing all the trophic process or all the interactions that occur there, it can be really interesting. Yeah.
And yeah, I was also, so besides the plankton communities, you might also consider, for example, plant pollinator data that are generated with eDNA.
Yeah.
Just you know, to really tease apart the effects of inferring your interaction networks and how this environmental DNA and molecular biology approach can affect, you know, the way you can infer your interactions, basically. And so I was wondering, yeah.
I think it's been reused. It produces a bit of different kind of networks, this kind of bipartite networks. So you simplify. But yeah, it would be really interesting. I think it can be done. So with Infinity, with this logical approach in general, you can have your rules of interaction. You can modify your hypothesis of interaction. And if you're able to express the abundance or presence data related with your rules, you can start testing also it on these systems. But the problem with the pollinators is that maybe your tool works well because you develop good rules and a good methodology to infer these bipartite networks for pollinators and flowers and our plant pollinators, let's say. But then this is not giving any insurance to then when you move to the microbial world, that then in the microbial world it will work. So it's a proof that the logic-based approach could work, but not that the logic-based approach for microorganisms could work, maybe.
Yeah, but maybe you could. So that's the second part of what I wanted to ask you is so. For me, DNA metabarcoding produces a lot of noise, and contrary to what you are suggesting sometimes in your manuscript, I don't think there is a consensus on the fact that metabarcoding produces very accurate lists of taxa because there is a lot of noise. And so in your manuscript, you considered abundance biases. But I think that even if you use data 2, so it's data 2, I don't know how to say in English. Even if you use this tool, you still can have a lot of false positives. So the errors, chimeras, and whatever. And so I was wondering whether, so to what extent your approach is robust to this kind of noise?
That's a good question. Well, we try to focus first on the most abundant ASVs, also to tackle a bit this, because as if you have a huge number of sequences, it's hardly to be more chimeric. We use also the up and downs for abundance. So even if you have ASVs that are not correctly classified, you can have problems, but then I also think that giving you some ecological information can also help you to understand what's happening there. And for the problems with abundance, we move from just correlation to up and downs, and I think this helps a bit because you are not using any measure of abundance. You just try to come out if the abundance is increasing or decreasing. So you also tackle some biases there on the abundance that they can be. So as we saw in the results, even with the real data, having an accuracy of 100%, it's impossible right now, I would say, but we have to start somewhere. And I think that waiting until we can do it perfectly, producing networks, it makes us lose time on other stuff. So if we can start producing networks with the most abundant ISVs, that we know that there can be some errors, but most of the information that we have there is interesting, it's still interesting to have them. Then we do direct interpretation. You need still ecology behind. And if we are with antagonists, which is a clear problem, and you detect some potential antagonists, you then will need to test them anyway on a lab, like ecoculture or this kind of experiments. But so you need to do ecology after and study networks, even they are interpreted. But using this methodology also helps you to do that, to figure out what's happening on the ecosystem. So you can say, okay, maybe here we have a problem. Maybe this ASV that we have here is wrong. It's not correctly sequenced, or this problem. Yeah, no, I agree with you. The problem is really complex and you can get lost, but the problem is not to try until you solve all the problems, I think.
No, of course. I mean, it's part of again. It's just a question, because I mean, if you are doing, if you are to use this kind of tools, for biomonitoring and decision for decision making, then it's, I mean, if you have errors in the data that might somehow, affect the patterns you are supposed to retrieve, then it's a problem, and so this needs to be addressed.
Sorry, I did all the metabarcoding process, and I found myself that all the problematics that you can be finding. We also had controls for positive, positive and negative controls for contamination, and this also, I think, that helped us to have some control this part on the molecular part of the procedure.
Yeah. So next on a more conceptual, and maybe it will be my last questions. So I was wondering, so part of your results suggests somehow that I mean, even if you can detect some environmental changes from several network metrics, let's say, it still somehow seems to perform less well than community level approaches. So it's my understanding from. So how do you explain that, and how could you improve your approach to better account for or to improve this detection of changes, basically?
So yeah, these diversity community metrics seem more great, because I think it's because when you build networks, as you say, there are many biases on sequencing, and you build networks, you add another layer of biases that it's introducing noise on it. So, and we saw it with the replical networks, and networks change a lot in each case. So there are really few consensus associations. So maybe it's the ecology itself. It's really, we have to understand exactly what an interaction network means, and maybe expect the same interaction network in the same ecosystem is wrong, and we need to have into account that each network will be really different. But we saw that they represent the ecological reality somehow because they change between cropping systems. But yeah, I think it's related with the network construction procedure and the metabarcoding procedure. If we give biased data to these diversity measures, we will have some problems, but maybe we differentiate better that if you give biased data to the network inference tools to then compare the networks. We have biases on the top of biases, so it's more difficult.
But I might be wrong because I'm not an expert on that, but I thought that in a complex system theories, when you have many interacting parts, it's difficult to make some predictions, and it's easier to do so on emerging properties of this network, or these interaction networks. And so I was wondering, what is the best level of aggregation of abstraction, that could help us to better monitor changes in ecological communities. If you have an idea of that, is it better to look at network properties or very big properties of the network, like modularity, just simple things, on the network, or even, or at the end, if it's not best to just look at the diversity of your community. So is it something?
Yeah, these properties like modularity of the networks seem not to reflect the ecosystem change for what we found. So we focus on this beta diversity to compare just the matrix of the network, let's say.
But maybe it's already too.
Yeah. That was done in this work is going from the 10% more abundant ISPs to all the ISPs that we have construct different networks and see if this was changing. So see if the interaction in the top 10% were more informative. And we found that at 10%, they were not necessarily more informative at 10%. So simplifying this complexity sometimes does not improve the results. No, no, it's really complicated. Yeah. But yeah, I mean, I think in this paper and in the whole work, we are not saying that using metabarcoding other than constructing networks is bad, or using networks is the right solution for everything.
No, I know. It's just
networks for most of the problems. So community measures can be perfect depending on your question or your ecological question on what you want to monitor. These diversity measures can be more, richness can be a great measure depending on what you want to monitor, for example.
Yeah, I mean, it's more from a practical perspective. I mean, these biomonitoring tools are developed, to be routinely used a lot by many people. And so, the network approach is quite complex. And so in this case, is it, necessarily the best solution, even if, I mean, let's say that you will improve it so that it is as predictive as the other approaches. Is it always still the most parsimonious way of, doing things, from a practical perspective?
Well, you're saying that improving it to 100%, I think this is quite really difficult right now. So we still haven't account that the network procedure have problems all the time. Yeah, as I said, it depends on your ecological question. I would try to decide it's the best methodology. Yeah, and when you select this, you have to take into account the biases. Even sequencing, just sequencing and just sequencing, doing sequencing and just metabarcoding, you also have some biases that you have to account for when you do it. And so you have to put in a balance and decide, depending on your question, what biases are you willing to accept and which no, so you can have to use another methodology.
Okay. Yeah, maybe I will leave the floor to Tristan. Thanks a lot for replies.
That's great. Thank you, Lucy. Tristan, if you'd like to.
Yeah. So thank you very much, Didac, for this nice presentation and your work. I think I will try not to second too much Lucy, but I think I would agree with most of what she says. That to be honest, I was a bit at first. I think it's a very ambitious work, and as a molecular ecologist, this interaction network is always a bit of the $1 million question that we would like to have, and that's super challenging. But I appreciated very much reading your manuscript for the same reason also that it was very clear and concise on what are the rationale and how when you conceptualize an interaction, what the effect of a species can have on another, how would something translate into abundance count and so on. And I think it was very well written and convincing. So that's very refreshing for me to have that kind of reading. The fact that you validated it on this Plasmoparaviticula pathobiome, I also find it very impressive and very nice. And I think it's a very a good first step to validate, even though I imagine, like as Lucy pointed out, to validate the tool on the other system other than microbial would be even better and stronger. So that's very nice. I found the discussion also very well structured, and you addressed a key question for monitoring that totally makes sense, and I think you address each of them in a convincing way, and that's even opening to some technical challenges and to some other question, like the way you suggest, like using co-culturing experiment to validate the approach. It was very nice because of course it's validating, but you directly also point to limitation of this approach and how we can we even make co-culturing that is convincingly reproducing what happened in the field. So yes, that was nice. I have also some question and comments. As a molecular and a DNA person, the part of your introduction where you present DNA amplification, sequencing, and so on, I found multiple unclear or unclarity there. So I made some comment on the manuscript that I won't get through that now, but I can send you after. Maybe one of the main questions that I have, and that I also felt during your presentation, is when you explain very well in your manuscript explainable machine learning, and you say that it is for model to be explainable, it's necessary to have first to introduce some scientific domain knowledge to the inference. And second, to make the inference process transparent and understandable. So while I agree very much with the second, that the first point that is a bit tricky for me, because I wonder whether by introducing some prior during the inference, if you are not a bit cheating to get the model validating what you expected to uncover. So that's in a way this question, if you think of it as the way you presented how you validate your tool by comparing afterwards to the literature, you come from both sides here on that particular question. So I wonder, what is your take on that?
Yeah, so I think you say cheating. At some point I had this not feeling of cheating, but I understand this point of view. But at the end, you want the network to reflect so you want the network to reflect the reality. And this is, in our case, the ecological reality, the interaction. So you at some point need to detect the interactions and need to define them before. So you're not cheating if you know that the network will just represent what you used on the rule. This means that if your rule is not right, the network won't be right, or will just represent, or you define just some kind of interactions and you miss other kinds of interactions, your network won't be represented. The whole ecosystem will just be representing that interaction. If you accept this, then I think the methodology is good and you're not cheating yourself. If you cheat yourself saying, I just provide a definition of something simple and I want to detect this simple thing, maybe you're cheating. But if you know what you're producing, I don't think it's a bad way. So potentially with explainable machine learning, what you do is this, but it's used in each system. In image recognition also, you can use it for recognizing faces. So you put some definitions on the shape of the nose, for example, or the shape of the eyes or the size of the ears, and then you look for them on your data. And then it's what your system does is this, okay, I differentiate this person from this person because the size of the nose is different. This makes the process explainable, and it's not cheating. It's you looking for difference on the face, describing what's the difference on the face for you, so you then obtain the explanation on why the faces are different.
So that may be a semantic issue, because when I said that you include some prior for the inference phase, it's actually more like you encode this as hypothesis that during the inference are kind of explored. That's a bit yeah. So that may be just a tricky line to make it clear, because for me it was a bit unclear for that. Then I have some question related to, I mean, when we read your manuscript and when we think of network from metabarcoding data, there is a lot of increase, decrease in abundance or that kind of things. And because metabarcoding is compositional by essence, that's always a bit tricky also in the wording. And so in some section of your work, I think it was very clear that you really state upfront that the compositionality issue in the data, but in some other part it feels a bit more unclear. And so related to that, I have some question, because I was surprised to see that you use this chi-square test of independence to infer differential abundant OTUs. So if I'm not right, to just say that, but there is many different tools that have been published for detecting change in abundance of OTUs between experimental treatments, such as DE-SIC2HR or so on. So I was just wondering why you used a different test for that, and if you would consider comparing your test with the chi-square with the other of this differential abundance test.
So I was using this because at the end it's the simplest solution, and we saw that it was working, so we keep it, because we just need to compare two samples. With this sec and this differential abundance, normally it gets more complex. So we checked it, we saw that it was working, and we decided to, statistically we decided that it was all right to do it. But yeah, you could use the sec actually. This is for deciding whether the up or downs are significant or no. So you could implement the sec to do this. It would take more time because the sec can be kind of slow depending on the size. But yeah, it could be implemented. It's a good thing. We thought we tested and we considered that our methodology was good enough to differentiate just up or down, significant up or down, taking into account the compositionality of the data. But yeah, it's actually, that's one of the good parts of our methodology. You could implement, instead of this chi-square test, a function for doing it from the sec. Just this will take longer to produce the data sets, and maybe test it and see if there is a big difference on the result, on the network result, or just compare the amount of up and downs that you get.
Then I have a follow-up question on that. Is that at some point in your manuscript you said that filtering of low abundance OTU is something that is common, because we all know that we have a distribution tail that is enormous, and most of the time, I mean, for like basic beta diversity, they are not informative. But I think in this context of network inference, I wonder, since the read counts are never independent for OTUs, how such a filtering, and you showed nicely that when you consider the most abundant OTUs and you include more and more rare, it changed a bit the result, but the trend remains the same. So would you advise, for example, what is your advice on this tail distribution of rare OTUs in data? Would you, I mean, I know it's improving the computational part, but do you think there is a value in keeping them for the network inference?
Yeah, so there is a divergence. There are papers suggesting to take them out, but there are others that show that some of these tail species can be really important for an ecosystem too. So if you take them out, you are missing information. So yeah, we took the most abundant because of computational reasons, but also because some of the less abundant, you have less information about them. because they are non-present on most of the samples. So you have all information about them being present in really few samples, 1, 2, 3 samples. And we consider that this is not informative enough to infer interactions using these species. And since we infer independent interactions, this does not affect the other interactions itself. And when we take them out after producing the up and down. So we never take out never modify the sequencing depth, let's say here.
So how can you keep sequencing depth while removing this?
It's as easy as you produce the logic clauses with all of them, and then you just select the logic clauses from the auto-use that you want to keep. Okay. So you have them on the data set at the beginning. You just take them out after. And the thing is that the abduction process works independently, never compares with a third species, or at least our simple design now is not doing it. So having them or not having them during the abduction is not modifying the result.
Okay, because that made me think about, you nicely say also that a 0 in a matrix, I mean, that's a challenge, and you showed that during your presentation also, because 0, you cannot say if it's a real absence or if that's a competitive exclusion. So, and you showed that for competitive exclusion, that was a bit tricky interaction to infer. So, yeah. So if you, intuitively, I mean, there is a direct impact of removing this rare auto-use for inferring such interaction. But if you say that they are still in the inference, again, I'm not an expert in there was so many technical details that I skipped, but
no, your question is really interesting, and it's this is at the end. The problem is you have really few information about this, about them. So, and it can be do many things like interactions and other. So with this few information, how can you be sure that the network inference will be good also? Try to take a bit of noise, even if you're losing information. Is this playing of, okay, how much noise I'm accepting to gain more information? So taking out them also avoid some kind of, you have less information, it's harder to infer interaction with less information normally. So you take them out and try to keep the ones that you have more information about to infer those interactions.
Okay. So maybe a last one. I'm very obsessed with compositionality, but yeah, I wondered, that's something that I didn't read also in the introduction, and I think that could have been relevant, is that's many different taxonomic group, and the copy number of ribosomal operon it can vary significantly between groups, and obviously that would affect your read count for taxonomic composition. So do you think, because I know that there is some databases mostly for 16S, so prokaryotes, but I imagine, and I guess you will agree, that including such prior knowledge or to make this read correction would improve the inference of interaction network.
Yeah, it will. Also, when we produce the up and downs, anyway, we just compare between the same ASV or the same O2U. So you want to know if the abundance of your O2U is increasing or decreasing. So you just compare their abundance. So this bias by the number of copies will be the same in all the samples.
Yeah, I see. Yeah. Very, very clear answer. Okay, I don't know if I use my 15 minutes. I have some other little, but
you have a few minutes more time if you'd like.
Okay.
Okay. Probably enough time for one more big question.
Okay. So then, do you have any idea why your method is less performant for, like, when you have less samples? You showed that at 20-30 samples, SparseCC and SpeakEasy are performing better. Is it a technical issue, or is it something that your method, I don't know, is learning less fast than SparseCC? If you give me the expression.
Yeah, it's how we describe the interaction. We have to look at different things. That we look at, for one species, how it increases and decreases between samples, and also if the other species causing this change, it's present or no. So we need to, on this system, we need to find examples of what's happening. So we need to look at this information and see how this is happening in our system. When we are around 20 samples, it's more difficult to find relevant information about this. So this compression measure that we obtain tends to be really inaccurate. So it needs more information to have this numeric measure accurate. But when we reach this amount of information, it reaches a plateau. So at the end, you are not increasing forever the accuracy. There is one moment that you have enough information to infer and you don't need more. But it's true that with 20, 30 samples, you are not in this case. You need information, or think another way of treating your data to make it more informative. We also played because we just use up and downs. With up and downs, you avoid some biases, but you are missing information because you are missing quantitative information here. So maybe we played with it and never found a good solution, but treat the information differently so you can extract more information, so be more accurate with less samples. It's one of the improvements that could be done.
Yeah. As you pointed, also you can include some qPCR. You did include some qPCR as a covariable also during the reference. Okay. Well, thank you. Thank you so much, Didac.
Thanks for your questions.
Thank you, Tristan. Christophe?
No?
Now.
It's work?
Yeah.
Okay, great. Thank you, Didac, for your presentation. I really appreciate it. And I have, as you can see in my report, more criticisms in the report because I have a little bit disappointed. There's a huge quantity of information, and you make a great work and trying to combine more theoretical, most experimental, and most mathematical or development of new tools. And so it makes sometimes the reading of your dissertation a little bit confusing at the moment. And perhaps this fact is due to that you want to present your dissertation as a succession of paper which are already accepted. So it's nice because we can see that the work is really appreciated by the community, but also it makes sometimes more difficult to see how you want to progress and how you ask the question and how you progress. And I really appreciate your talk. And if you could, or if you have the ability to translate your manuscript with the feel you try to use in your talk, it's really very nice because your talk is very, very, very good at the moment. So congratulations for that. Try to make this effort and to present your work in a more linear fashion that in the dissertations, which is a little more complex for me to follow. Anyway, I think you address a major question for people working on interaction, microbial interaction, or more generally in organisms in interaction in the real life. And so I really appreciate to see the science is moving, because as microbial ecologist, what I already know is try to make some correlation, to try to infer some interaction at the moment. And so to see that some people try to develop some new tools and to try to be less correlative and try to put in biological information in our ability to represent the network of interaction is very, very appreciated, I think, and it's a strong impact in our scientific community to have these tools to try to have a better understanding of microbial interaction. So congratulations for that, and to your supervisor to give you opportunity to develop this work, because I think it's make a good improvement in the scientific community at the moment. I have a lot of questions I already asked, but I want to go back to your model you use, because I think that to address this question, working on microbial community is very hard topics to do that. And perhaps as Lucy asked you to work on other organisms, and perhaps to work with organisms where we have more information at the biological point of view about the interaction, like insect community, for example, could be a very interesting thing to do to improve or to test your method and to see if you are very confident with that or not. Because I think that microorganisms are particular organisms It's not evident to make a clear correlation between the species and their functions. And so, okay, we are able to see some change in composition of interaction, but if you want to really make a biomonitoring of ecosystem in changing global change or this kind of thing, I think we need to have to put some more functional aspect in the interactions that just to see some difference in the species abundance. What is your feeling about that?
Yeah, I think you are right. Working with the animal systems or arthropods, insects, it could be really interesting because it's something that we know better, so we can establish better rules for these interactions. As said on my example on the presentation, there is this size thing, there is many these mouth parts, there is many things that we can use that we know better. We can even observe directly the interactions, which makes real clear validation. If you say this is this, and then you're able to put them together and see one eating the other is good. But so yeah, would be really good to validate. But the problem it's on the edge of, okay, I validated with this animal system, so it works, but with my rules for animals, with my way of obtaining the abundance data, but this does not ensure that then it will work on the microbial world. So, and that's the problem. And if I move back to the microbial world, I will have the similar problems. So I took this development of these logic pairs, trying to establish a basis, even if it's not perfect at all and needs some improvement, but start having some facts about microbial community. We can be sure about this. And from there, start improving this. Our approach has room for improvement in many ways. We can introduce this relation with databases. I say it a lot. I played with it, but never ended testing it also because of the problem with databases. But on these databases, we can have a lot of functional information about microorganisms. Like there is Faprotax, fungild. We can know if fungi is a saprotroph or it's a parasite and this kind of stuff. And we can also include, so we can use also during the inference or as testing to start to be sure on how the microbial interaction works.
And you are confident with this point because when you say, for example, we know that this fungi is perhaps I don't know, a saprophyte fungi, for example. We know that it's based on taxonomic informations and what we know from an ecological point of view. But as you know, in microorganisms, there is a not directly relationship between the species and the function. So, and perhaps with the same species, we have different or completely opposite issue in the interaction, no?
Yeah. And I stopped this part because also the databases are quite incomplete and not really correct itself. So yeah, it's a problem. But somehow it's this, we need to start studying microorganisms. So we need to start putting a basis on how we study these interactions, combining with the interaction like lab experiments could be good, because could be also a good basis to start doing these culture experiments and start developing a frame to test interactions, could combine with the interaction network inference from many data could be interesting also.
Okay.
So yeah, I think it's test. I really want to, as I said, Dave has some data set with arthropods that can be really interesting for testing. I'm really interested on doing this and would be really good to see that it really works there. But I also think that some microbial problems are really microbial, so you have to start working with them to solve them. That molecular systems for some of the stuff cannot be really useful, maybe.
Okay. And so working with microorganisms, a lot of people say when we work on microbial ecology that we make a lot of correlation. And so we use a lot of statistical approach to try to make sense for our data. And so as you know, there is something we try to move now. It's try to connect more ecological approach for microbial ecology with most mechanistic people working more on mechanisms in microbial interaction. And so people will classically work on microbe interaction. So with a dual interaction, it's very easy to follow and to analyze from a mechanistical point of view. try to complexify this interaction and try to use some synthetic community approach to try to converge information, so to reduce the complexity of markable community to something from human people which are perhaps more easy to manipulate and to understand. So do you think, for example, that working with synthetic community, I think about the work from people from the Max Planck, Stefan Acker and other people like this, we try to associate or to develop synthetic community approaches with 100 or several hundred bacterial species and some fungi which are already in interaction. Do we think it could be a good approach? And if yes, what kind of experiment you could propose or you could imagine to consolidate infinity mathematical development you make to go more in detail in interaction network?
So I think that, yeah, this work with mock communities could be a really, as I said, we don't have good testing data sets. So being able to produce yourself good testing data sets. And as I said, for example, we've seen that we try to model different interactions. If we can produce some model data set with some interactions that you really know that happen, some of no, or some other data set where you know all the interactions, these kind of models, of course, it helps improvement. Also, if you're able to make synthetically some kinds of interaction, you can see if you are modeling this type of interaction explicitly, correctly or no. So yeah, I would sign now to have this kind of data sets at the beginning of my PhD and being able to produce them. But still, with this link between lab work, let's say, and the network inference, there is always the question of, okay, what I'm producing in the lab are the necessary conditions for interaction that will happen then in the ecosystem I want to monitor. For example, on the leaf, most interactions can be due to space, basically. So there is a fungi there, so the pathogen cannot go inside the leaf because the another fungi is occupying the space. And this condition is you are able to reproduce it on your synthetic data. So even if synthetic data can be really interesting, still you are not really sure on you are reproducing the correct conditions on the lab.
Okay. And so with your most experimental work, so you work on fungal community, on fungi. In ecosystem functioning, we know that fungi is one part. There is some bacterial community, which are their own microbe interaction. And in fact, there is also some interaction between the fungal and bacterial community. So it's a more complex picture we have in the real life. Do you think, or how do you imagine the improvement or the development we need perhaps to do to try to put some more complex information to build the network? So perhaps both to compile bacterial and fungal information from metabarcoding. And so for you, it's very easy, just another OTU or ISV, and so you just increase the size of the table, or you need to put more other information, or you need to make some other development to try to do that. If I want to use your tool, for example, with both bacterial and fungal data, how in practice I do? And
so for the metabarcoding datasets that these papers are on the thesis, we have also the bacterial part. So you have the fungi, we have the bacterial data, but now the bacterial data is bigger and maybe not so important in some cases on some of the ecological context. So we focused on the fungi, but we have the bacterial. And yeah, I would say that I think that now in Infinity you can provide two different O2U matrices, and you will produce these abundance clauses from each matrix and then just join them. So as a starting point, I would do that. It's one of the things I'd like to do. I haven't had time yet in the thesis, but in the future it would be really nice to build a common network and then try to figure out if it's working or not. And from there, you can still introduce logic rules if you can figure out on how bacteria and fungi will interact between them, and maybe establishing rules that I don't know, because you know that bacteria fungi interactions would be the same as fungi or bacteria interactions. Yeah. You need another rule for this kind, so you can make the model more complex. But yeah, first I would try directly and then go to more complexity from there.
Okay. And do you imagine some some complexity in the ability to make the output of the results due to computation aspect, or because, as you say, you try to work with fungi because it's less complex. So if you use more complex ecosystem, do you imagine it to have some calculation limit or this kind of thing? And you indicate that you need to improve, and you always make this development to have something which are more rapid in terms of computational aspect.
Yeah, we are, as I said, the abduction base is done with Pybel, this program developed by our colleagues in Surrey, and it works better, but still, yeah, when you reach some size of information, it can take a lot of time. And also it uses a lot of memory, because on how abduction works, you need to produce format the data in a way that occupies a lot of memory. So, but it's a working process, and they are really good and really clever. So I hope in the future, both things will be developed in parallel. So the implementation of logic machine learning will allow you to start introducing more and more data, which is a bit limiting now. We can do it with fungi ecosystems, maybe with some part of the bacteria communities, but still, yeah, we need some work on it. And maybe from the part of the implementation also, we could do some improvements to make it go faster and be able to manage this. But for now, it can reach a point either where you don't have enough memory on your computer, or the execution time can increase a lot. So it's not useful to wait for 2 weeks for having a network.
Okay. If I have 2 other questions, I have time. Okay, thanks. So the other question I have is, you use a particular ecosystem data set for your experimental approach, because it's a pathogenic system. So you have the microbial community, you have the host, you have interaction between microbe, but also with microbe and host. And you have in a disease process, when you make your observation, you compare healthy and disease tissue, if I well understand the process, and you try to see some difference between these both aspects. So in fact, you have a very contrasted ecosystem between a healthy system and disease system. And how do you increase or how do you take in account in your analysis the interaction with the host, which potentially could be another factor we could monitor or could impact indirectly the composition of your community, and so the abundance between the different OTU, which are not directly related to microbe interaction.
Yeah, it's something to introduce to. We haven't done it because we were considering that the biotic interactions itself so there is this thing of how you consider the infection, and a biotic pressure or a biotic pressure because of the increase on abundance of the pathogen. So it's now, let's say that we treat it like a biotic process. So the changes that can be produced in the microbial community are mostly caused by the development of the pathogen. But yeah, in the future, we know that the plant, when has some lesions, would throw some metabolites to the context. So having this information, and I think it can be introduced in the logic model, can help you to better model this pathogenic system.
Okay, and so with your development, can we use some temporal data, for example, and it's very easy to improve, to increment in the model. If I have a kinetics, for example, from a healthy tissue, when I inoculate the pathogen and I want to monitor what is the evolution of the composition and the interaction before to go at the end of with a disease tissue, with a lesion, or with a plant which are in disease context. It's possible to make this kinetics, or you make a succession of network, or you have the ability to integrate the dynamics in the model.
So now, yeah, it's maybe what we do is maybe cross-sectional. So you need several samples from the same moment to build that network and maybe compare it. But is this flexibility that I was talking about, if we can come out in a theory on how an interaction goes a long time. So how we, in these logical clauses, we will have another factor, which is time. And in our theory of interaction, we have to describe what is the effect of time in interactions. But if we do that, we could use temporal data. I haven't done it because I was working with cross-sectional data, and maybe for biomonitoring, it was more interesting to capture at one point in time a network and then see if this network changes in another point in time, then just check how an interaction can evolve during time.
Okay.
But yeah, it's about having this your hypothesis of interaction, and then format the data. The abundance changes would be the same. You just will include the time on your data and try to make an hypothesis of interaction. So we can sit and talk about this if you want at some point.
Okay, thank you. So I have finished. Congratulations again, Didac, for your presentation, for all your work you do. I think you make a significant improvement for our community with Infinity, and I am very excited to use it with my own data, because I think it's yes, very relevant. I think to make a most logic systems that just correlation, which need, and I think we could make some. To ask or to put some good hypotheses to be tested from an experimental point of view when you have this kind of output from our data. So congratulations again, and good luck for the rest of your research aspect.
Thank you.
Thank you.
That's great. Thank you, Christoph. So Didac, thank you very much. I really enjoyed your talk. I very much enjoyed reading your PhD thesis as well. You've got access to the report I wrote on it, so you can see my comments in there. I share the positivity of the rest of the committee in terms of all the good things you've done. I do see a great advance in a very difficult and complex aspect of science. So I think this is really good to see. I also am suffering here from being the last person to ask questions from a panel of extremely expert researchers, and as of such, I've been slowly ticking off the questions that I was going to ask that other people have asked before me, and whittling down to what I'm left with. But I do agree heavily with everything the committee has said, and particularly some of the methodological details that Lucy and Tristan raised about the inherent nature of meta-barcoding data. And I think you've done a good job addressing them, but I think it's certainly considerations to move forward. So without trying to repeat anybody's questions, I'm going to ask a few technical questions. And then I thought this might happen, so I had a load of backup questions to do with the computational side of this that I will ask as well. So first of all, and this is sort of, the flavor of this question is very much related to stuff that Lucy and Tristan were talking about. And it's to do with the way the data you use for Infinity are processed. Okay, so you use the Data2 pipeline. It's one of many. It is currently the most popular used pipeline, but it's certainly one of many. And even within Data2, there are loads of different choices that you could have that would affect how your data looks at the end of it. And so I'm sort of curious as to what properties, or whether you've looked in this, what properties of the meta-barcoding data most cause differences within your network, your inferred networks, and which parts of those sort of properties of the data are the things most acceptable to people's choices in the bioinformatics flow. So I'm curious as to, so imagine you've got somebody taking one data set and person analyzes it one way, they make a certain set of choices with their bioinformatics, another person analyzes it a slightly different way. Those two data sets will be slightly different. What differences in them is Infinity E most susceptible to causing changes in inference from? Or have you not really looked at this yet?
No, because our idea was to try to do a pipeline that mostly can use any kind of meta-barcoding data. So we tried to generalize this problem, tackle them together more than going to that. As we said, I'm thinking, sorry, but as we compute the abundance changes, it requires to have good. It's good if the sequencing depth is not crazy changing. For example, this can help a lot to have a more clear up and downs and assess better the significance of the abundant changes. And also, it's about how many species you select or ISVs you select. because at the end, the problem is you need that each row or column, so the auto-use on your matrix are really functional units. And this, it's a bit, it also can depend on how the pipeline you follow, on how you group the sequences and which sequence you discard. If it's really you're grouping a functional unit together, so you can build a network in based of that. Because we've seen that sometimes some ASBs can be from really similar species and have the same behavior. So at the end, there you're losing some information because your pipeline is giving you, it looks like it's giving you two functional units that should be one. We try to address that by clustering it with Lulu, doing a post-hoc clustering a bit to improve the resolution, but still from if you look at the taxonomic classification and the alignment of the sequences, we still have some ASBs that could cause some noise because they could be from the same functional unit sometimes.
Okay, that makes sense. So, I mean, I've got another question that's sort of slightly more philosophical in nature. So a bit more of a thinker as opposed to anything to do with the methods or the exact analysis. But are we holding ourselves to hostage here by trying to infer interactions that have been defined for macroorganisms on microbial data sets? So you think about the things you've talked about competition, commensalism, mutualisms, blah, blah, blah. The majority of these interactions have been really well defined on macroorganisms. And as we move down the taxonomic chain, fungi are weird things in themselves. And then as we start to move into prokaryotes, it gets even odder. Are we soldering ourselves here to sort of ransom in trying to infer interactions that are not necessarily well defined for the microbial systems you're looking at anyway?
Yes, actually, we stayed with this because it was simple, and we need at the beginning we need some kind of simplicity to start looking at this. So yeah, definitely. We are. The problem is, how do you find the correct definition for these interactions? I think the good point is looking for these simple interactions you can understand, and then from there try to study them, maybe do some lab experiments, testing them, and try to refine from them. But this, at least, even if it's not the best description of interaction, it can describe a group of interaction or can describe factor of the interactions, for example, so you can at least have some information that you can use to develop your knowledge on interactions. And then, of course, in a future, your logic inference will have this complex definition of interaction. But we need to figure out which complex interactions are there. So we need to start testing somewhere. So we started for these simple definitions. Then, of course, it's good to check and see if we can go more complex from there.
Okay, that makes perfect sense, and you know, it is a bit of a deep thinking question, but the idea is that you've now got these interactions, and then some metrics of those networks can be used to infer something about the ecosystem they come from, and more importantly, changes in that ecosystem if we're looking at biomonitoring perspective. And I was wondering about this because in one of your earlier chapters, you were showing that the sort of standard community level metrics, so species richness, community composition, those sort of things, they differ between your different cropping systems, and the networks that were inferred from them didn't differ as much. Okay, and this got me thinking because the data is being analyzed. There's a couple of differences between the way those two data sets are being analyzed, one of which is the spatial scale they're being analyzed at. So if you imagine it, you've got microbes on leaves and the average diversity of microbes on leaves, but the first network will be across all those leaves at the plot level. So you've got leaf versus plot level. So your network is working at a different spatial scale than your community composition data. And also linked to that, so not only have you got spatial scale changing here, but you've also got the degrees of freedom. You've got fewer replicates of networks than you have of leaves, for example. So for a given biological effect size, you're always going to find a more significant result in the community composition of the community data, because degrees of freedom and p-values scale together, than you do with the network sort of side of things. So I'm sort of curious, I guess the question really is what is the appropriate spatial scale and level of replication for using networks in biomonitoring? And is it surprising at all that we see differences between community type data and network type data when we're looking to infer changes in an ecosystem?
Well, there is many questions here. Now, the first thing is, yeah, of course, you have more degrees of freedom with the community analysis with the network. But if you want to monitor, biomonitor, the network can provide you some information that the community-based analysis does not, which is the biotic interaction. And this can help you then to take decisions. So even if you can be less inaccurate and you have more degrees of freedom, it can be, if you show that it can be used for biomonitoring, then for making managerial decisions, you will have another tool that you can use complementary to the ecological analysis. And related with the sampling size, there is a big discussion on the sampling size that needs to be used for building networks. There is people happy with 20 samples. There is others that say that should be 200. And I think it's still related with this absence of data to be able to test properly if what we are getting is good enough with the number of samples we have. So it would be good to have some kind of MOX data set with different number of samples where we can start really testing the sampling effect. Because I tested with the synthetic data, and for the correlation tools, it does not have a really huge effect, more in infinite. But it's something that it would be really good to test in any tool, the real effect on your sampling design. Because as you said, as you have more degrees of freedom on the networks, but at some point you also may expect some kind of plateau on your accuracy differentiating the ecosystems. And this would be the number of samples where you want to focus. And it would be really interesting to design experiments to figure out what's the number of samples you would need for that.
Yeah, I mean that makes perfect sense. I mean, I'm just curious. Imagine a network. The number of nodes within a network scales with space. Yeah, that's your species area relationship. Do your number of edges scale accordingly, or do they have a different scaling property? I don't know what the answer is here, by the way. I'm just curious.
Yeah, it depends. No, mathematically, it could increase the number of edges. Increases exponentially with the number of nodes, because each node can potentially have an interaction with all the other nodes present. So if you do randomly a network that with a random number of edges, as you have nodes, it's exponentially the probabilities. In reality, it's not that much. You should respect that it's not that much, because not all the species interact with all the other ones. You expect subgroups in a network, and so for what we've seen is that when you increase the number of nodes, the edges increase quite linearly with it.
Okay. Oh, that's interesting. That's interesting. Okay, so I'm going to just change my tact a little bit on the questions. I've got a couple of minutes. And I'm curious about the Infinity R package. And I'm curious about this for a number of reasons, one of which is because I really want to use it and see it get into the public domain. So I'm curious as to a couple of things. I mean, what improvements do you still need to make on that? Do you think is required to really get that absolutely ready to be rolled out? Start with that one.
So, sorry, can you repeat the question?
Are there any improvements or methodological changes to the Infinity R package that you've been working on that need to happen before you can really release it to the world?
Yeah, the problem we have is we use Pygol for the abduction, this other program. This has been newly developed, and they are really interested in protecting it and being able and having the right license and this stuff. So the way we're using it now, it makes it kind of difficult to compile and use in all computers. And this is something that we are working on now, because for now it works Infinity in some Linux computers, but I think in Windows machines would be really difficult right now to work or to configure it to work. So this is a step, as we can get a more easy code to use for Infinity, would make a good improvement for sharing it. And for the other stuff, the package is there. I use it myself, but it needs a lot of beta testing, because I've been using for given data sets. So the next poor person that uses it will find problems, errors, and because for now needs testing. So it's good But if I'm a bit afraid of sharing it to everybody because a lot of errors will emerge because it needs testing with other kind of data sets and improvements.
I mean, is there any sort of computational limits to it at the moment, or is it reasonably okay to handling the size? I mean, because the problem with meta-barcoding data is every year it increases by an order of magnitude in terms of size.
No, now it's really limiting. Now we have to focus in most abundant ISVs and not huge sample numbers. So it's the first limiting stage. It's something that when you use it, you have to take into account. If you go to big data sets, it can take a lot of time. That's why so during my talk and on the manuscript also I say that depending on the kind of networks you want to obtain or the kind of monitoring you want to do, maybe correlation tools, if you have these big data sets, can be more interesting because it gives you a speed, and this speed allows you to test and fail and this kind of stuff. For now, the most limiting part is the execution time and memory usage.
Yeah, that makes sense. Okay, so that's my time up for questions. Dave, you've got your hand raised.
If I'm allowed to respond to just something that Didac said, I would like to, if that's possible.
Yeah, you can respond, please.
No, it was just Didac said that he was afraid of other people using it. Actually, we're not afraid. He's afraid of the amount of work that it might imply in him supporting it. We actually want people to use it, and we very much encourage people to use it. But under caveat that he's just finished his PhD, he doesn't have a job to go to, and if anyone does happen to have some money, he'd love to take that money off their hands and maybe support more people more widely.
Thank you for that, Dave. That's, I think, a perfectly acceptable interjection to try and plug somebody getting employment and doesn't affect the committee's deliberations at all. And certainly something we can discuss offline. Okay. That's all the questions for the committee. I'm just going to do a quick check for the committee members to see if there's anything that has been raised that anybody wants to follow up on. We all okay? Yep. Okay, that's wonderful. So now what's going to happen is we are going to leave you, Didac, and are going to deliberate and decide on stuff, and then we will return to you with our verdict shortly.
Okay.
Okay, so I'll see everybody later. For the panel, I'll send around a Zoom link via email in a second to join us on, okay? All right.
Thanks.
Thank you, Didac.
So 1 second. Okay.
Hey there, Didac.
Hi, hey, yeah. Hello.
So we're waiting for everyone to come back. They should all be joining shortly. And Dave is currently
Here.
there. Oh, okay. I couldn't see. I thought you might still be running, Dave.
Yeah, it sounds like it actually. My heart's going, but no, I made it. So how does it feel like doing a PhD Viva from your back room?
Oh, it's good. New way of working, isn't it?
Yeah.
Yeah, it's different. It is different. But to be fair, I think over the last few years, this is the fourth or fifth I've done from here. So
Okay.
the difference being I've not done one before where there's a huge lecture theatre in front of me with people sitting in it. That's the slight difference.
Everyone took the opportunity to go to the toilet and grab a coffee.
After 2 hours and a half, something like that.
It's not.
Just a small comment. I remember while reading your manuscript, I kept wondering, how do you pronounce your package name? So I assume it was Infinity, but it was like
It is Infinity, but it gets pronounced as Infinite, but it's supposed to be Infinity. And yeah,
It's me. At the beginning, I was calling it Infinite, and now my brain sometimes just plays against me. And I say it Infinite, but it's Infinity.
I must admit, Tristan, I had exactly the same thoughts.
Yeah. It's always important to have a very catchy name for a new
Oh, yes.
software. Yeah.
That's it.
it's a pity you won't be here for a beer, Alex.
No, I know.
Yeah.
I'll get drunk at this end on your behalf, Dave.
Yeah, drunk in your broom cupboard there. Yeah.
Well, when I go to the pub tonight and tell my wife it's for work, you know.
What's it like in Norway at the moment, Tristan? Is it dark?
Yeah, now the sun is setting at 3:30, so we have a bit less than 6 hours of daylight. That will be yeah, like that for another couple of months. It will start to increase again, of course, but yeah, it's a bit tough. That's the moment of the year that is a bit tough.
Have you taken to the traditional northern approach to actually coping with lack of daylight, which is to drink basically all the time?
Yeah, I do that, and I also drink some. So I also take some of these pills of D vitamin. I really don't want to experience the symptoms of lack of daylight depression and related. So I'm trying to be preventive. Yeah. No, but it's beautiful. We have snow since this morning, so now that's looked nice on a mountain. Like that.
Have they got lost, do we reckon?
Maybe they are in the other Zoom meeting.
There was a few links, and I clicked on I thought that was a link that I came in first, and it was not working anymore. But by trying 2 links that were saying it's expired, I arrived here, and there you are. So I imagine they might be doing the same.
Okay, I'll try again from my end.
Here.
Here's Christophe. if Lucy isn't able to join us in the next 2 or 3 minutes, should we just do it anyway? Because it was unanimous.
Yeah, we can crack on. Let me just forward the link to Lucy, just in case that's the issue.
Aha, here goes.
Precious.
All right, wonderful. Everyone's back. Okay, I'm going to reconvene this defence. So, Didac, it is of great pleasure that I'd like to let you know that the committee is unanimously recommending the conference of the title of PhD in Population Ecology on you. And we will be providing that recommendation going forward. It's worth noting that we have no corrections for you to make to your PhD thesis. We thought it was excellent. And we would like to congratulate you on producing a very robust and clear defence of your PhD research and all the hard work that has gone in behind that to make that research possible to be presented today. So well done. Big congratulations from us. Dr. Didac. Excellent. Well done. So I presume Dave is now going to take you out and get you utterly smashed, which would be entirely appropriate. I don't know if anybody else on the committee wants to say anything.
No, you said it all. It's a big congratulations to you, Didac, and I wish you the best for the next step.
Yeah.
Yeah, thank you very much. And thank you very much for being here in this defense. It's been really nice. Thanks.
It's a pleasure.
More than welcome. Dave?
Yes.
So I presume that's it. I don't want to hold up the celebrations any further. I think you guys should go out and celebrate wholeheartedly now. Is it okay that we bring this to a close now?
Yeah, you guys log off. We're going to
Okay.
Yeah, thank you very much, guys. Really, it was great talking to you. The questions were great. It's made a lot easier by Didac actually doing a really great defense. Talking personally, it's been a great 3 years. I mean, apart from the fact that they locked us up for something like 2.5 years with this excuse called COVID, it's really, really good fun. I've enjoyed working.
Yeah, it's been really nice 3 years. I really enjoyed doing this thesis.
And all that. Thank you for your time, guys.
No, you're more than welcome. Go and celebrate.
Thank you
Dave,
so much.
just a question, Dave. Do you have a Twitter account or something like this? Do you have a Twitter account, or because if you just post some picture about the celebration, it would be great for us. Or just send the picture by email or something like this.
Bye bye. See you.
Enjoy your time. Bye bye.
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 Thank you.
Thanks.